Pleasure to have with us today Fernando Pineda. Fernando is a good friend. We go fishing once in a while, and I've known him for many years. He started as a physicist. He's a physicist, in fact, a theoretical physicist from College Park, one of the best physics PhD programs in the country. And left, okay. And then he decided that machine learning was really the thing that was interesting. He published the first recurrent backpropagation paper in PhysRev Letters, in the late 80s, in the heydays of neural networks. So this is the Pineda of the recurrent backpropagation. If you've probably seen that paper. And then he went to APL, where he did various things, including acoustic processing and even VLSI. At one time, we're actually working together on some VLSI project. The last 6 years, he took a plunge into the world of bioinformatics, and that plunge landed him at downtown in the School of Public Health, where he's in the Department of Biology and Microbiology and Immunology. And he's going to be talking to us today about his recent work in that area. Okay, there you go.
Okay, so this talk is a bit different from your usual talks in this seminar series. So what I will be doing is providing you with a brief overview, if I can get this to wake up. All right. First, before the overview, the acknowledgments. So this work is done with a number of people. In particular, Sammy Huang, who was a computer science undergraduate here, was instrumental in helping me write some of the pilot software. Gang Wu was a postdoc in my group for a year. And Nirbhay Kumar and his lab are malaria parasite experts, and they actually provide me with actual parasites. The Malaria Research Institute gene array facility is where we performed microarray experiments to confirm some of our detections. Karen Beeman is in the biology department here on campus, and we're collaborating right now to understand what, to learn to experimentally characterize some of our detections. And finally, Alain Sewer provided us with some of his code very early on in the project, and we wouldn't have been able to even get started without it. We've moved beyond it now, but those are the people who have helped. And why won't this advance? There we go. So I'll start with a brief introduction to malaria, so why this problem is important, and also a bit about the parasite that causes the disease. Then we'll do a molecular biology 101, which is kind of a quick trip through the salient parts of the molecular biology that you need to understand what the problem is. Then I'll briefly discuss some computational aspects about RNA gene finding, which is already a big field, so I'm just going to scratch the surface and mostly talk about what I've been doing. And then I'll discuss a pilot study and some results from the pilot study, and also finally with what we learned algorithmically in the pilot study, and how we're already changing the algorithms for the next generation. And finally, I'll make a pitch for any students that might be interested in working on this project. So malaria is a disease caused by a protozoan parasite. That's a one-celled organism, and it's spread by mosquitoes. Approximately 40% of the world's population is at risk, so this is a major disease. 300 to 500 clinical cases each year, and there's 1 to 1.5 million deaths each year, most of which are in children under 5 years of age. And most of those fatalities are caused by the Plasmodium falciparum species. So there's different species of this parasite that cause different variants of malaria, but most of the fatalities come from Plasmodium falciparum. And as you can see, it's endemic mostly in sub-Saharan Africa, the tropical areas on the globe. The common feature here being that these are very poor areas. There used to be malaria in Baltimore, but it was eradicated just through good public health. So here's the life cycle, simplified life cycle. Okay, so it's a parasite that's carried in mosquitoes. When the mosquito bites the human, it releases sporozoites, which are one stage of the parasite. These invade the liver. It takes about an hour to get there, and they invade liver cells where they reproduce. And in that stage, they spend a week to 10 days. When they mature, they rupture the liver cell where they've been maturing and go into the blood system, where each of the individual merozoite stages will find a red blood cell. It'll invade the red blood cell and go through a cycle of maturation there. Cycles are about 24 to 48 to 72 hours, depending on the particular species, and those cycles are associated with fevers that people that have the disease have. So after one cycle, somewhere between 1,000 and 10,000 new parasites are released from a blood cell that ruptures, and they in turn infect other blood cells. And this repeats. Okay, it doesn't grow exponentially, except in severely immune-compromised individuals. But there's a stage there of asexual reproduction. Okay? Now, a certain fraction of these parasites don't go into this stage, but instead mature into sexual stages. You know, the analog of sperms and eggs, right? Those so-called gametocytes are also released into the bloodstream, but they don't reinfect red blood cells. What happens is that they hang around waiting for a mosquito to bite, and so they're ingested into the mosquito, where the male and female gametocytes mature, become gametes, and join to become a new organism. That organism then penetrates the wall of the midgut, the stomach of the mosquito, and finds its way into the salivary gland of the mosquito, and then the cycle repeats. Okay, so this organism basically has 3 stages, right? It's a liver stage, a blood stage, and a mosquito stage, all happening at the mosquito stage at a totally different temperature. And this simple outline is actually simple compared to what's actually happening. Okay, so there's a blood stage which is quite complicated. There's the there's the liver stage. There's the blood stage. There's the sexual stage, and the mosquito stage. And if you were to look at the parasite in each of these stages, it looks like a different animal. it goes through developmental stages, kind of like an insect undergoes metamorphosis, right? Metamorphosis, I mean, caterpillar, pupae, mature adult, right? 3 stages. Well, this has, you know, I'm not sure how many stages are here, but there's a lot, okay? So there's a lot of complexity in the development of this organism. And approximately 80% of the genes in this organism are turned on and off in a stage-specific manner, okay? That's a lot compared to, say, humans or mice or rabbits or anything like that. So it's a huge fraction of the genes which are being turned on and off. And there's many lines of evidence which point to unusual gene regulation mechanisms in this organism. And in particular, the evidence seems to be pointing towards non-coding RNA. Okay. So next question, what is non-coding RNA? So to do non-coding RNA, we need the biology 101. So we'll start with DNA, which is the famous double helix, which is a polymer. Okay, so one strand of DNA is a polymer, which is just a long molecule composed of 4 repeating subunits. Okay, let's not worry about chemically what these 4 subunits are. Let's just call them A, T, C, and G. And they repeat in some order, which conveys a message or messages on this polymer. So the messages which are on this polymer are everything needed to build and operate an organism, be it me or a parasite or whatever. So here's what a DNA molecule looks like. Okay, it's 2 strands, and a strand is not symmetric. It has a direction that's determined by chemistry, and these 2 strands, they line up in an anti-parallel fashion, and the bases pair up. Okay, so there's so-called bases here. There's 4 kinds of bases: cytosine, adenine, thymine, and guanine. And A's always pair with T's, C's always pair with G's. Okay, so that's DNA. It's a very stable molecule. It's so stable, in fact, that you can buy a beer now that's been brewed from yeast that was extracted from an insect that was extracted from 25-million-year-old amber. Okay, so it's I think it's called fossil beer. So this configuration is very stable. All right. So here's that DNA molecule reduced just to its sequences. Just reduced to a symbol sequence. There's a so-called sense strand and an anti-sense strand. Now, there's something called the fundamental dogma of molecular biology, which is how you get from this DNA sequence into proteins. And proteins are what the most molecular machines in your cells are composed of. Okay, so here's what, this is just terminology right now, okay? So you've got the DNA, which is a 4-letter code, A, T, C, and G. That DNA is transcribed into a single-stranded RNA. Now, RNA is a molecule very much like DNA. It's just that there's just one oxygen missing it has an extra oxygen in a part of the backbone. So that has a 4-letter code, A, U, C, G. And usually that's just a temporary message which is used in the cell. This message is read and used to create proteins. Now, proteins are also polymers composed of amino acids, and there's 20 amino acids in these polymers. And basically, each triplet in the RNA corresponds to one amino acid. Okay, so this particular sequence then corresponds to a particular sequence of amino acids in a protein. Okay, so here's a protein sequence. It's a one-dimensional polymer. Okay, so what happens then in the cell? This one-dimensional polymer folds up. Okay, just folds up and becomes a functional machine in the cell. Okay, so all protein enzymes, all these machines in the cell are composed of folded up proteins or folded up RNA. Okay, so just to give this a little life, where's the mouse? Okay, so this is an animation of DNA double helix right in the middle there. You can see the base pairs forming plates. Okay, so that is used. So each of those DNA molecules is then transcribed. Okay, here's a DNA molecule. And what you're seeing is an RNA polymerase machine self-assembling on the DNA. And once it's completely self-assembled, it'll take off. Watch. Okay? So it's taking off, and it's reading that DNA and sucking in single nucleotides and spewing out a strand of RNA. Okay, there's the RNA. So this RNA is then exported out of the nucleus, and it creates, and what happens, another machine called a ribosome finds it, and then starts marching along, a triplet at a time, putting together the protein sequence. Okay, so that's the so-called dogma of molecular biology. So now, what's on this DNA sequence? What you can see here, this is one chromosome of the Plasmodium parasite laid out, and there's regions that are coding for those proteins. Okay, and they can be read in either direction. But notice that there's also a lot of stuff in between. Okay, in between those protein coding pieces, and that's not converted into protein, but it is transcribed, although it used to be thought of as being junk sequence. In fact, it probably does something. So here, this is the fraction of a genome in various organisms that codes for proteins. So in E. coli, which is a lowly bacterium, about 80% of the DNA sequence codes for proteins, and everything else is non-coding. As you move up in complexity or sophistication, this is yeast, a worm, fruit fly, mouse, and humans. What is it that you see? You see less and less protein coding sequence, and apparently more and more junk DNA, which is kind of odd. In fact, recent experiments show that almost all this junk DNA is in fact transcribed into RNA. So cells are using a lot of effort to process this, to convert it into RNA. So it must be doing something. And it's believed that non-coding RNA has regulatory functions. All right, so now we're particularly interested in regulation by non-coding RNA in Plasmodium falciparum. So the genome of that organism was sequenced in 2002. There's about 5,000 protein coding genes. About 50% of the genome is protein coding. The rest is non-coding. Now, one of the very bizarre things about this organism is that in most organisms, there's a balance between C's and G's and A's and T's. It's like about 50% C's and G's and 50% A's and T's. Okay? In this organism, the coding regions are about 23% A's and T's. Okay? So if you were to calculate the entropy of this, it would be much lower than a typical genome. Worse yet, if you look at the intergenic regions, okay, the intergenic regions, the regions between the genes, the AT content gets down to 13%. Okay? So it's very low complexity stuff. Okay? So that causes problems with gene finders, and it causes problems with sequencing as well. So there's emerging evidence now that what? I'm sorry.
On the chart that you showed there, which is on the previous page, I mean just
the one before.
Colorful chart that you showed. So where does this? So this will fall somewhere.
Yeah, it falls about here.
Just right.
Yeah. So between yeast and worm.
I see.
Okay.
So between a worm and yeast.
Yeah. Yeah. Okay, so there's emerging evidence now that this regulatory RNA is involved in pretty much every single step of RNA processing and control of regulation. So this is a story that has evolved in biology over the past 10 to 15 years.
So just to define the language, because
yeah,
biologists
it's all language. Yeah.
Regularly means that something that is really
controlled
by the environment.
No. So
depends on the environment.
No. So something has to control. It's the mechanisms inside the cell which are controlling the production of protein.
I see.
For example, yeah.
That's called regulation.
That's called regulation. Okay. And that can be in response to environmental stimuli, but it could also be in response to, it has to go into the next developmental stage, okay, to differentiate. Okay, so let's talk about computational gene finding. So the function of RNAs typically depends on their structure. Okay, so yes, they're non-coding RNAs are single molecules, but they tend to fold back on themselves and pair up. Right, the base pairs pair up. So here's a fold. So gene finding algorithms in general tend to use secondary structure prediction as a starting point. One way of describing this fold is with a so-called, I'm not sure what this is called technically, but it's dots and parentheses. It's a balanced parentheses expression. And a dot means an unpaired base, and a parentheses means a base that's paired with a partner. Okay? And starting from the outside in, you can see who's paired with whom. Okay? So those of you that like strings like this kind of thing. Those of you that like regular languages don't like this kind of thing.
How do you know that they are paired?
Well, you don't. I mean, you to determine the structure requires a structure prediction algorithm. Okay, and there's no guarantee that that's working properly. Okay, so what's the general idea of these? What you try to do is you try to minimize the Gibbs free energy of a given structure, and you can imagine you try all possible combinations, but of course there's a dynamic programming algorithm that does that, and basically it takes into consideration the contributions from the energy contributions from the base pairing. It also takes into consideration the energy that comes from stacking these plates on top of each other. So in particular, the particular sequence here, AU on top of AU has a different energy from AU on top of GC. And takes into account loops and stuff. So that's an order n cubed algorithm. If this and that's if this is a can be defined with a balanced parentheses expression. There's also pseudoknots which are allowed, in which case the folding problem is NP hard, but with heuristics it can be reduced down to n to the fifth or n to the fourth.
Question: Are you minimized? This is in 2 dimensions or 3 dimensions?
So this is in 2. Yeah, exactly.
Is there a difference as it would apply to 3 dimensions?
Yeah. So you can't have pseudoknots, for example, in 2. Yeah. So this is just what's called the. So the primary structure is just this particular sequence. The secondary structure is what you're seeing here. The tertiary structure is when the whole thing folds in 3D. Okay. So a very common kind of structure in RNAs are so-called hairpins, where there's a stem and a loop. Sometimes there's bubbles. So these happen to be hairpins from so-called microRNA precursors. Here you have the secondary structure of ribosomal RNA. And what you can see is that there are many long hairpins. Okay? Of course, they have bubbles in them a lot.
So the reason that you see a lot of those things is because they are thermodynamically stable.
Yeah. So basically, they pair up and then they kind of wind around and form a little helix. And it's just a.
They allow it for more, I guess, complex structure while stable.
Yeah. I mean, I don't know the exact reason, but it's just a commonly seen structure. So that's what we're going to be looking for. Okay. More generally, there's a very powerful conservation-based approach for finding general non-coding RNAs. And here's what you do. You take sequences, DNA sequences, from different organisms, kind of the same region, and you try to align them. Okay? And then once they're aligned, you try to find a fold for the consensus. Okay? So this is like C, C, G. Well, that's not a good one. I don't know. Yeah, A, C, A. You might say, well, that's about an A. So you treat that as an A. Okay. So then you try to fold the consensus sequence. So this one, winds up folding like that. You also wind up scoring things like mutations. Okay. You can't just mutate one position and maintain the fold, right? You would have to mutate, if this C mutated into an A, then this G would have to mutate into a U. Okay. Okay. So there's software for doing that. It's been published. It's quite powerful. The only problem is it's only sensitive to RNA whose primary sequence is evolutionarily conserved. Okay. So you wouldn't think that's so bad. Except that there's evidence that many non-coding RNAs are evolving very rapidly. So sequence conservation isn't such a wonderful thing, right? It would be nice if you could detect RNA without non-coding RNA without using sequence conservation. So it turns out that's been done in the case of herpes viruses and also microRNAs in small stretches of DNA. So here's the basic idea. you start with some intergenic sequences, which are reasonably short. You then fold those short sequences. You actually do this in a sliding window, and I'll explain why that's done. You then try to identify the folds, the secondary structure in those sequences that are robust. By robust, I mean it folds the same way independently of the flanking regions. Turns out that that's a good principle for detecting biologically relevant sequence. But even if you do that, you still get an awful lot of false positives. So some filtering is involved, and then what comes out is your non-coding RNA. Okay, so if you can make that work, then you can find RNA that's evolving very fast. Okay, so here's more detail on that approach. Basically, you take a window, and in that window you apply this RNA fold, one of these folding algorithms, and you get the secondary structure. And then you record which pairs matched up with which nucleotide matched up with which other nucleotide. Okay, and then you slide the window over, fold it again, and now you build up a histogram of how many times each nucleotide paired up with each other nucleotide. Okay, so kind of pair statistics. And then Sewer calculates this robustness factor, which is basically the number of windows containing a given pair divided by the total number of windows that contains those two nucleotides. Okay? And then he looks for robust pairs, and he looks for robust secondary structure, which kind of has the flavor of the dynamic programming algorithm. He says, okay, if I have a secondary structure, then the secondary substructure of that hairpin must also be robust, and et cetera, et cetera. The problem is I don't think he knew about dynamic programming, so his algorithm is kind of a hack. But the first time we did this, we just used his code. Okay. Okay, so here's what happened the first time we did this. We found a lot of hairpins. Okay, so this is chromosome 1 to chromosome 14. Plasmodium has 14 chromosomes. And for stupid reasons, we only scanned in one direction. We didn't scan in both directions, which we really should do. So this is the number of hairpins. So how many of these hairpins are real, and how many should you expect to find by chance? Well, you can answer the question, how many you expect to find by chance, by shuffling your sequences and reapplying the algorithm and seeing how many hairpins you get. Okay? The only subtlety is that you don't simply want to permute all the base pairs. What you want to do is a special permutation that permutes pairs. Okay? It's called an Altschul-Erichsen shuffling, and basically it preserves the dinucleotide frequency, okay? Not just the mononucleotide frequency. And the reason you do that is because you have this base stacking contribution to the energy of the stem. So who follows whom matters. So you want to preserve that in your null model. So when you do this is what you get. Okay, you get essentially the same number of hairpins. Okay, so most of what you're detecting is junk. Okay, it's false positives. It's not biologically relevant. What?
It's random.
It's random, exactly.
Not necessarily junk.
Well, so it's not biologically relevant. It's an artifact of your algorithm.
Okay, that's what you mean.
Yeah, that's what I mean. Okay. Okay. So we somehow need to filter these, and these are the kinds of things that you find. Okay. These all look perfectly plausible as biological hairpins. So here's what we did. We took three features. So the base composition. So remember,
Fernando, just again
yeah,
to bring things in context.
yes,
So really, this is all about string matching.
string matching.
String, I mean,
Yeah,
this-
I mean it's strings. We're all working in the world of strings now. Yeah,
That's it.
yeah. We left.
With 4 letters, and that's it.
That's it. Exactly. Okay. So which is why I'm talking about this here. Strings. Okay. So base composition. So in this hairpin, you can calculate what percentage of the symbols are G's or C's. Okay. And you know that the intergenic region, it's what, 13%? It actually gets down to 10% in some places. Well, there are papers that say that if you make a threshold at 35% in the intergenic region, you can actually identify regions that have non-coding RNAs. It actually works. Okay. Well, we said let's set that to 15%. Okay, so just a little bit above random, yet lower than the 35% that is in some of these papers. Okay, so our other threshold was thermodynamic. Our other feature was thermodynamic stability. So basically the energy of the fold. And finally we looked at dinucleotide Shannon information, which is just it's just the information content of the dinucleotide pairs. So each hairpin then can be represented as a point in a 3-dimensional space. What we can do, we can take our shuffled sequence, right, and plot all those points in this space. Okay, so we know that the region where all those points are, that's where all the false positives are. Anything that's substantially outside that region is something we should be looking at. It's novel. Okay, so basically that's what we do. So this is just a 2-dimensional projection. This is C. elegans, it's worm, another organism. And there we ran this algorithm on a couple of the chromosomes in that algorithm. And then we also plotted where the known microRNA precursors would lie. Okay, so this is the energy here, and this is the Shannon information. So this is the false positive region, and this is the true positive region. You can see most of the bang for your buck here is actually coming from just this thermodynamic stability, right? But there's a bit of a contribution coming from the complexity, the Shannon information. Right. The third dimension. Okay. So if we apply this technique now to each of the 14 chromosomes of the organism, okay. So here's one chromosome here. So this is the energy. This is the Shannon information.
So what happened to the complexity, to the chemical complexity, or the third feature?
Yeah. So it's okay. So it's sticking out. So GC content is sticking out of the board, and so if we cut that at 15%, everything above 15% is colored red. Okay. So the blue region is the false positive region. All these grays, reds, and what's under here is our detections from the unshuffled, and these red ones are 15%. Okay, so if we cut at 50% GC content, and these are just kind of arbitrary lines that we did. Again, this was a pilot study. We didn't use any sophisticated false discovery rate control or anything like that. We just said about there, about like that. Okay, and the reason we did that was we wanted to select just 10 candidates because we figured that's all we could experimentally validate. Okay, so we just made things very stringent. Okay, so so we got 82 candidates. Of those, here's some of the interesting ones. So this is a very nice hairpin that we found. that's the hairpin. That's where it is. It's oriented this way. And when we did a blast search at GenBank, we came up with a hit, which was this sequence here, which was annotated as non-coding RNA. So we were jumping all up and down because this wasn't a published non-coding RNA. The sequence had been deposited with just that statement, but nothing had been published on it. So we contacted those guys, and they said, yes, we're investigating that non-coding RNA, and they immediately rushed out a poster because they were afraid that we were going to scoop them. But we're not laboratory-based scientists, so there was no chance of that. But we talked to them. But this was the first evidence that we had that we were actually on the right track for detecting non-coding RNA. Here's another one. Looks like this. It points this way, which is antisense to this this gene for adenyl cyclase, which is actually involved in regulation in cells. Okay? So we're collaborating with the Beeman lab to try to characterize this particular hairpin. I might add, we detect the hairpin, but the non-coding RNA is probably much longer. So we're collaborating with the Beeman lab to find out how long that is and what its function might be.
That's why the previous sequence was so big.
Yeah, right. Yeah, exactly. Because they detected it some other way and then done the lab work to find out how big it was, and then they had deposited that sequence. And when we blasted and searched, we just our matched here. Okay, so the next thing that we did, we had these 82 hairpins, which were like between 80 to 150 long, nucleotides long, and we did a number of experimental and database searches to see if they could be real. Okay, so one thing is that you can search so-called EST databases. So that's express sequence tag. It's where an experimentalist has tried to experimentally determine all the RNAs that might be expressed in a given sample. So in fact, we got a bunch of hits there. So those were positive detections. We also did what's called RT-PCR, and we confirmed a number of detections. And finally, we made a microarray, which also confirmed a number of these. Okay. So overall, 25 of our 82 candidates are actual transcribed RNA. Okay. So this is a good thing. We think that we're on the right track, and now we need to make this a better algorithm. Okay. So what are the algorithmic issues? Okay, so the pair statistics where we fold in windows, it's computationally a very intensive thing. So we basically did the same thing that Sewer did. We took the software called RNA fold, and we folded in a sliding window. Okay, but that's the complexity is L cubed, where L is the length of the window, and N is the length of the sequence. And basically, that's the most computationally intensive part of the calculation. Okay? It takes 2 to 6 hours, depending on which chromosome you're working on. In the same library, there's another package called RNA pfold, which instead of doing the folding, it actually calculates the pair statistics for you. And because it's not doing folding, I mean, I'm not exactly sure of the algorithm, but it actually has much lower complexity. Okay? So that's what we're going to do. We're going to use RNA pfold in order to get these pair statistics directly without doing the folding.
Excuse me, L is typically
500. Okay. Oh, and that's a good point. Because this is only L squared, conversely, we can either keep L the same and go faster, or get the same speed and use a larger L, right? Which would actually be the more interesting thing. All right. So once you got these pair statistics, we basically used Sewer's code to find the robust hairpins. And when the first time with SAMI, with Sam Huang, that we tried a longer sequence, we said, okay, well, let's toss in the whole chromosome instead of just a small segment. We ran it for a day, 2 days, 3 days. Finally, we figured out it wasn't going to stop. Okay, so we didn't bother to actually see what he was. Well, we did look at what he was doing, and it wasn't pretty. So what we did then is that I just implemented the dynamic programming algorithm to find robust hairpins, okay, from these pairwise statistics, and now that's very fast. This used to be very slow. Okay, next thing, we don't exploit thermodynamic stability relative to random sequence. So it turns out that it's been going back and forth now, but in many papers, but it's now pretty much nailed that that if you compare the thermodynamic stability of an actual structural RNA compared to the thermodynamic stability of that same RNA, but shuffled, so shuffle it, fold it, shuffle it, fold it, shuffle it, fold it, you'll get a distribution of energies. Okay? Well, for non-coding RNA, the actual RNA is much more stable than all those shuffled sequences. But for messenger RNA, I think ribosomal RNA, for random RNA, it's not more stable. Okay? So it's a good signal for pulling it out. The problem is you have to do all those samplings and reshufflings and refoldings, and you need about 1,000 of them. So it's not really applicable on a genome-wide scale. So fortunately, this problem was partially solved in a code called RNA-Z, but they only applied it to mononucleotide shuffling, so permuting. And they actually stated in another paper that they couldn't figure out how to do it for dinucleotide shuffling. So we figured out how to do that. It was actually just a simple matter of fixing how they train their algorithm. So we've done that. So now we're using that. And finally, false positive rejection. As I showed you, it's very ad hoc. We'd like to use more formal methods, and that's just a matter of using more. Pardon? Oh, I'm sorry. Support vector regression. Yeah. So here's our second generation pipeline. This exists. So you take the entire genome and you do three things. You just take each chromosome, okay, call that the Watson strand, and you also take the reverse complement of that strand because you actually need to search it in both directions because you don't because genes can be encoded either this way or that way. Okay, and the third thing we do is we do a dinucleotide shuffle of the genome. Okay. All right. Then for each of those three versions of the genome, we apply the following algorithm. First, we apply we calculate the pair statistics. Okay, currently using the sliding window approach, but eventually using RNA p-fold. Then we reconstruct. In other words, look for robust hairpins using our dynamic programming algorithm. Then, once we've got robust hairpins, we featureize them. Okay, we calculate their energy, their z-score, their GC content, all a list of features. So then we have a list of hairpins on the forward strand, hairpins on the reverse strand, and then we use the false positive hairpins to determine the false positive regions, find the outliers, and those are our non-coding RNA candidates. Okay? So that's what we're proposing to do. We've actually done this to some extent. I'm going to skip over this. So we've done this. So this is what happens when your postdoc leaves at a critical time. The only plots you have are on paper. So what this is the dinucleotide Z-score. This is the GC content. Okay. So on this plot, if we take all the hairpins that we found in the false positive, in the shuffled sequence, basically 99.99% or some fraction are in this contour. Okay. So these are the outliers. That's what we should be looking at for real non-coding RNA. And you can see here, this is a nice example because there's basically two sets. Okay. There's the set that have high Z-score, large negative Z-score, okay, which is, papers out there saying that's what you should be looking for. And there's a set that have high GC content. Okay. Okay. So, and then we find a bunch out here. So once we get this up and running, these will be the candidates that we will be looking at and studying much more intensely. So the conclusions are, an initial approach is unbiased against evolutionary conserved sequence, so we can find rapidly evolving non-coding RNA. We've shown it's feasible and that we can detect real transcripts. The full extent of the transcripts has to be validated, has to be determined, and we also need to validate additional transcripts. And we're collaborating with the Beeman lab. We've shown how to scale up this approach. Okay, our pilot scan took a lot of computing, and it was kind of dumb and not very scalable. But we've solved pretty much, I think, all the computational bottlenecks. And now it's just a matter of having some bright young student that wants to work with me to do the remaining 5%, which of course is 95% of the work. Thank you.
Any questions? Fernando, I have another question.
Yeah.
So, you talk about features, and features here are kind of funny because there is thermodynamic physics fixtures and information fixtures.
Yeah.
And the third one was kind of chemistry, structure.
Yeah, exactly. Kind of the hairpin, right?
Yeah.
Well, so there's GC content, there's Shannon information, which actually we're not going to be using in the future. I don't think it gives us anything really.
I see. Which is interesting. So it's really all either physics or chemistry that is really giving you any
and structure, right? The first scan is structure, so we're looking for hairpins.
structure.
Yeah, robust hairpins. That's kind of the first cut.
Mm-hmm.
Okay. Then after you've got robust hairpins, then you featureize them, and right,
You look at, okay.
and then you reject all your false positive hairpins.
One question. You apply the dynamic programming approach upon the hairpins that you find initially. Is that right? Finding areas with
Upon the pair, yeah. So on the pair statistics, right?
Of the hairpins, though.
No. So what you've got is think of it as a two-dimensional array of I and J. So how many times does nucleotide I pair up with nucleotide J? So now you have to do a little dynamic programming algorithm in that matrix in order to find hairpins, robust hairpins. So hairpins that are formed from pairs that have, and are and have high frequency, high probability. So each element in each value in that matrix is basically the number of windows that the number of sliding windows that a pair occurred in. Yeah. Yeah.
I just had a quick question. So you had mentioned before that you ran the windows sliding one direction, you didn't run it sliding the other direction.
Correct.
Although that could have given some useful results. Why
Right.
would that have changed
Right. So when I started working on this, I didn't think it would matter at all. And then, of course, if I'd been an RNA biologist, I would have known that was bogus. So oops, going the wrong way here. These are all the extra slides, sorry. Let me find a match here. Okay, so in DNA, A's always pair with T's, and C's always pair with G's. So if you were to make a loop of DNA and you reverse complemented it, you would get the same loop back. Okay, because just all the A's would change to G's and all the G's, right? So you would get exactly the same. So secondary structure in DNA is invariant with respect to reverse complementation. But RNA has a different problem. A's will pair with U's, C's will pair with G's, but C's will also pair with U's. Okay, and if you reverse complement C's and U's, or is it G's and U's? Well, I don't know, whichever it is. If you reverse complement that, the reverse complement of that pair doesn't pair. Okay, so in other words, if you take an RNA structure, an RNA sequence, you fold it, you get a fold. Now take the reverse complement, fold it, you get a different fold. Okay. Yeah.
So in the first instance, you have 4 simple letters, but they only compare pairwise. It does. In the second instance, you can actually.
There's kind of a mixing term between the
between the two.
Yeah. Right. Yes.
So for the non-biologically inclined amongst us, can you give a little bit of overview what kinds of experimental confirmations will now be? So you have this candidate of non-coding regions and all
right.
that are potentially biologically significant. What
Right.
kinds of things do we find when we study non-coding regions?
Well, okay. So there's 2 questions there. So if we have a candidate, say, how do we confirm it's actually in the cell? So there's a number of ways of doing that. One is by using a technology called reverse transcription polymerase chain reaction. And basically what you can do, the polymerase, you know, that enzyme that goes running along DNA, spewing forth an RNA loop. No, I'm sorry, that was RNA polymerase. DNA polymerase is an enzyme that will walk along, reverse transcriptase, I'll get this right, is an enzyme that will walk along an RNA strand and put down a complementary strand of DNA. Okay, so now you have an RNA DNA hybrid. Okay, but in order to get that reaction started, you need to attach a primer. So here's your RNA. You need to attach a primer. Okay, so and once that primer is there, the reverse transcriptase will start elongating. Okay, so the trick is to get a primer that's complementary to some part of your hairpin. Okay? So if that hairpin is actually floating around in the cell, the primer will find it, and then the reverse, this happens in the test tube, right? The primer will find it, and then when you attach the reverse then the transcriptase, reverse transcriptase will find it and elongate the DNA, then you'll get an RNA DNA double strand. And then you have another primer that, you know, one primer's here, another primer's here. You heat the whole thing, does this, and then you put a second primer, reverse primer here, and then you use DNA polymerase, which then starts elongating this way. and then you get these two strands of DNA and DNA. Okay? Then you heat again, those separate. Now those two primers that were there before find those partners again, and then each of those individuals get two more, right? And then you heat again, those separate. Now each of those 4 individuals turn into 8, 16, okay? So after 2 to the 10 cycles, right, you've amplified by 1,000, and you've only amplified the RNA that matches your primers. Okay? So you design your primers to match only the RNA you're trying to find. So once you got that amplified, then you run that out on a gel, which is, it's like chromatography, right? Where you basically length matches into position on the gel. And if you have an amplified product, it shows up as a band. If nothing was amplified, there's nothing there. So.
Okay, so I think maybe we should just finish. And
Yeah.
if someone wants to